Hello everyone, my name is F. Arioga, and today I'll be going over the prompt for the advanced concepts presentation, where I'll walk you guys through how pyruvate is harvested and then converted into acetyl-CoA. So let's get into it. Now, of course, the conversion of pyruvate to acetyl-CoA is a crucial step in the cellular metabolism that bridges the gap between glycolysis and the citric acid cycle. It takes place in the mitochondria of the eukaryotic cells, where the pyruvate generated during glycolysis is transported from the cytoplasm. Now, of course, the conversion process involves multiple enzymatic reactions that produces acetyl-CoA, and that is a key molecule that serves as a substrate for the citric acid cycle. So the very first step is pyruvate decarboxylation. The conversion of pyruvate to acetyl-CoA begins with pyruvate decarboxylation. The step occurs in the mitochondrial matrix. The enzyme responsible for this conversion in pyruvate dehydrogenase complex, also known as the PDC. The PDC consists of three main components: the pyruvate dehydrogenase E1, dihydrolipomide transactylase E2, or otherwise known as E2, and dihydrolipomide dehydrogenase E3. So of course, pyruvate hydrogenase catalyzes the oxidative decarboxylation of pyruvate, resulting in the formation of an acetyl group. In this process, one molecule of CO is released and NAD+ is reduced to NADH. For E2, the acetyl group generated in the previous step is transferred to the lipoamide cofactor attached to E2. This forms acetyl dihydrolipoamide. Dihydrolipoamide dehydrogenase, otherwise known as E3. For this step, FAD accepts the hydride ion from the dihydrolipoamide, generating FADH2. The lipoamide cofactor is then regenerated, and NAD+ is again reduced to NADH. And then step 2, the formation of acetyl-CoA. The acetyl dihydrolipoamide produced in the previous step is then further processed to generate the acetyl-CoA molecule. So of course, the acetyl the first step within this is the acetyl transfer. Coenzyme A, also known as CoA-SH, replaces the lipoamide cofactor, resulting in the formation of acetyl-CoA, and the thiol, or the SH group of CoA, forms a thioester bond with the acetyl group. And then we go on to the regeneration of the lipoamide cofactor. For this step, the E2 subunit of the pyruvate dehydrogenase complex transfers to the acetyl group of CoA, regenerating the lipoamide cofactor. The lipoamide can now accept another acetyl group from the next pyruvate molecule. And then next we go on to cofactors and energy carriers. During this process, cofactors such as NAD+ and FAD are reduced to NADH and FADH2, NADH2. These energy carriers will later participate in ATP synthesis during oxidative phosphorylation. And of course, at the end of this process, one molecule of pyruvate is converted into one molecule of acetyl-CoA, and the remaining steps of the citric acid cycle, also known as the Krebs cycle, occur in the mitochondrial matrix, where the acetyl-CoA enters to undergo further metabolism and energy production.